Thank you, Steve. Welcome, everyone, to Aspen Brain Institute's Expert Series 2.0. Welcome to our growing community of global seekers, seeking to be educated and delighted at the cutting edge. Hello to people all over the US and 103 countries who have registered for this series. Thank you all for tuning in. We are offering our new free series in order to build an online global community focused on brain health. We'd like to thank Lugano Diamonds for their generous support as our national underwriter. And we thank Lugano again for choosing Aspen Brain Institute as a nonprofit worthy of their support and their values, and for making our Zoom expert series free to the public and able to be viewed worldwide. We also thank Alpine Bank for their generous sponsorship over many years now. And they're the best community bank in Aspen. And we thank our partners at Brain Futures who are doing important work in advancing the practical application of new scientific understanding of the brain and learning in K-8 schools. At our expert series, you will hear global leaders, deep thinkers, technology experts, top-notch doctors, and extraordinary creatives. We hope their insights will transform how you take care of your brain today in a way that your 80-year-old brain will thank you for tomorrow. The Expert Series 2.0 underscores Aspen Brain Institute's mission to share access to the top minds and evidence-based research on brain health, also to increase brain health literacy globally, and ultimately to create a brain healthy planet. That's our plan. It is my honor and privilege to bring you these new episodes. Let's see what we can learn and explore together. And now to our expert series. I want to introduce Dr. Ronnie Stangler, Chair of Aspen Brain Institute Scientific Advisory Council. Dr. Stangler is physician and psychiatrist and clinical professor at the University of Washington and co-founder of Genome Advisory. Ronnie's going to introduce our next expert speaker, Dr. Morgan Levine. Ronnie, take it away.
Glenda, thank you so much. It is truly an honor to introduce you to Dr. Morgan Levine. Dr. Levine is assistant professor in the Department of Pathology at the Yale School of Medicine and a member of both the Yale Combined Program in Computational Biology and Bioinformatics and the Yale Center for Research on Aging. What Yale tells us about her work, and this is a direct quote, Dr. Levine utilizes an interdisciplinary approach, integrating theories and methods from statistical genetics, computational biology, mathematical demography to develop biomarkers of aging using high-dimensional omics data. She has extensive experience using systems level and machine learning approaches to track epigenetic, transcriptomic, and proteomic changes with aging and incorporates this information to develop measures of risk stratification for major chronic diseases such as cancer and Alzheimer's. Her work also involves development of systems level outcome measures of aging aimed at facilitating evaluation for geroprotective interventions. A number of the existing biologic aging methods that she has developed are being applied in both basic and observational research, meaning it's complicated. Please, nobody stop listening now, because when I asked Morgan what she does, she responded, and this is also a direct quote, "I help create great tools that people can use to live healthier, longer lives." After receiving her baccalaureate and doctoral degrees from the University of Southern California, she conducted her postdoctoral research at the Department of Genetics at UCLA, where she partnered with Dr. Steve Hovarth, a pioneer in development of the biologic clock. She was then invited to join the faculty at the Yale School of Medicine. Please know there are several different groundbreaking versions of biologic clocks. One of the first was, of course, the Hovarth clock, and another is often called the Levine clock. Today, Dr. Levine will discuss what your epigenetic profile reveals about your personal aging. In her talk, she will define epigenetics. She will more accurately define age. We'll address how tracking biologic age can help us optimize our health, as well as discuss epigenetic science aimed at literally changing the pace of our aging. On a very personal note, we are especially proud to introduce Dr. Levine to our Aspen Brain Institute community, as she truly represents the next generation of great young scientists, and a female scientist at that. Remember her name, please. You will be hearing about her extraordinary work for years to come. Dr. Levine.
Thank you, Dr. Stengler, and thank you to the Aspen Brain Institute for inviting me. I'm really excited today. And as Ronnie alluded, I'm going to really try and break this down, because my ultimate goal is to have the work that we do be usable by individuals in the community to better their health and aging. So today I'm going to talk about what we like to call your true age. And basically that boils down to what our epigenetics can actually reveal about how we're truly aging on a biological level. So one of the most exciting things about living in this time and the technology that's being developed is that we're better able to track and monitor a variety of different things about our health. We can track our steps, our exercise, our heart rate. We can take EKGs on our watches. We can look at our genetics, track our sleep. And now there's even contact lenses that can track glucose levels, which is important for diabetics. But what I would argue is that we're actually not tracking the one thing that's probably has the biggest impact on our overall health, especially down the road. So I usually like to start when I'm teaching some of the classes at Yale or in some of my talks, asking the question, kind of, what is the biggest risk factor for lung cancer? And of course, you guys can't answer given the webinar, but a lot of people will say something like smoking. So we know that smoking can increase lung cancer incidence and death by about 15 to 30-fold when you compare smokers to non-smokers. But in all actuality, aging is actually the biggest risk factor for something like lung cancer. So at the age of 25 to 29, you have about a 1 in 200,000 chance of developing lung cancer. But by the time you're in your 70s, this now increases to about 800 in the 200,000. So you have an 800-fold increase for people who are 75 to 79 versus 25 to 29. Same thing can be said for other major diseases. So this is showing Alzheimer's and just the prevalence of Alzheimer's in the population as a function of age. So as you can see, in people under 65, while there are a few cases, it's very rare to actually see Alzheimer's disease, and most of these are familial-based Alzheimer's. But as you get up into your late 80s, you can actually see that the prevalence of Alzheimer's disease is actually quite high. But at the same time, not everyone who grows old will develop each of these age-related diseases. So they show some of the major age-related diseases, so cardiovascular disease, cancer, Alzheimer's, COPD, and diabetes. And as you can see, basically this empty spot above are the proportion of people who do not develop these diseases during their lifetime. So there's actually a large proportion of people who will not go on to develop these diseases. So whether you're going to develop one of these diseases or not, a lot of people might think could come down to chance. But in reality, it's actually biology. We don't think that it's just your probability with time is going to increase and some people are unlucky and some people are lucky. We actually think that the changes happening on a molecular or biological level are actually what are giving rise to these diseases. And we call this biological aging. And then the next question is, can we actually measure this biological aging process? Number one, to figure out if we can intervene in it, or number two, just to figure out our own personal risk and to be able to actually promote some sort of health promotion and or disease prevention strategies. So when I talk about biological age, I like to give kind of the analogy of a race. So let's imagine that everyone is running a 10-mile race. So even though everyone might start the race at the same time, if we stopped it, let's say after 10 minutes, we might see that some people are only a mile into the race, whereas other people might be 2 miles into the race. And so this is kind of the same concept of biological age. We follow people for a certain amount of chronological time, akin to the 10 minutes, but then we can actually see what proportion of this race have they run? How far are they through what we might expect their lifespan to look like? And as you can imagine, this is not a race you want to win. You want to be the slow and steady one in this case. So one of the ways that we actually have been measuring biological age is using something called epigenetics. And specifically, we're focusing on one form of epigenetics called DNA methylation. So basically what that is that your DNA gets wrapped up and it has these chemical modifications to it, which can change the conformation of how the DNA is wrapped. So the epigenetic or DNA methylation is basically a chemical tag on your DNA. When it's there, the DNA wraps more tightly, and when it's not there, it's looser, and you actually can express genes located in that area. And this is really important because, as you probably know, nearly all the cells in your body have the exact same DNA, except perhaps from some mutations you've accumulated. But what makes a stem cell, a blood cell, or a nerve cell is this epigenetic code. So the DNA methylation and other epigenetic factors actually change your DNA structure, and this changes the phenotypes expressed by the different cells. So I like to think of epigenetics and specifically methylation as a recipe. So let's say your genetics are similar to all of the ingredients you're going to ever need to make anything you can imagine. And the epigenetics or DNA methylation is the recipe that each cell will use. So every cell might have a slightly different recipe that they're going to use to make whatever their phenotype is. But one thing we know is that these methylation patterns or epigenetic patterns seem to change with age. So much like you might have your grandmother's old recipe written on one of these cards, over time, what's written there becomes faded or not clear, and you might actually not be reading it correctly, which could produce an aberrant cell type. So the way this kind of looks actually in the genome is, again, this is your DNA, and we have these regions called CPG sites, where basically, if you look from what we call a 5' end to a 3' end, so you're just following one strand down, you have a C or cytosine followed by a G or guanine. And these cytosines have a propensity to become methylated. So these are these chemical tags getting added to them. But the interesting thing is, for many of these, what we see is that the number of, or the proportion of cells in your body that have these chemical tags changes with age. So let's say we took a blood sample from someone and they're 20 years old, and we find that 5% of their cells in that sample are methylated at this specific region. And then we look at an 80-year-old and we find all of a sudden now 45% of them are methylated at this region. You could also look at a different region, and perhaps in the young person, the majority of cells, so 90%, are methylated in that region, whereas in an 80-year-old, this reduces down to 60%. And these changes in your epigenetics and your DNA methylation actually have implications for your health and your overall phenotype. So because of these very consistent and predictable epigenetic patterns that happen with age, we've actually been able to develop what we call epigenetic age measures. So basically what we do is we can take a sample, whether it be a blood sample, a tissue sample, a skin sample, a saliva sample. We can sequence the methylation, so we can actually measure the methylation levels in that sample at up to 850,000 sites across your entire genome. And then we use different machine learning techniques to actually develop these epigenetic age measures. So you might take samples from all these 35-year-olds, and we find that even though chronologically they're for 35, maybe one of them is biological age of 31, another might be 37, and so on. And the very interesting thing is that these epigenetic age measures don't just predict someone's age very accurately, but divergence between your chronological age and your epigenetic age is actually a very good indicator of your future health and disease risks. So when we measure these in blood, we find that people who are epigenetically older than expected based on their chronological age tend to have an increased risk of breast cancer in the future, lung cancer, heart disease. They tend to have shorter telomere length, for those of you familiar with that. We can look at individuals whose parents are centenarians, and they tend to have lower epigenetic ages. We also find that epigenetic age is associated with things like cognitive impairment and the number of diseases an individual has. But to me, one of the really exciting things about epigenetic age is that you don't just measure it for the whole organism or in just a blood sample. You can actually measure it in essentially any tissue or cell type in the body. So this is just an example of 3 different tissue and cell types. So we can measure it in blood. And again, we find it tracks with age, in skin, in breast tissue, and in brain. And basically, we can also see that not all your tissues tend to age at the same rate, but we actually see some samples age faster than others. And actually, when we look at this measure in brain specifically, we find that your risk or the likelihood that an individual had Alzheimer's disease is also shown in their epigenetic age. So this is just showing the chronological age of individuals, and this is their epigenetic age measured in their actual brain sample. So this is post-mortem after death. And again, when we look at your relative epigenetic age to your chronological age, so in this case, higher means that you're epigenetically older than you are chronologically, we find that people who have higher epigenetic age tend to be more likely to have had Alzheimer's based on neuropathology, and also to have presented with cognitive deficits akin to dementia prior to dying. Another exciting thing is we can also develop cellular specific aging measures. So your cells themselves actually age and have different lifespans. And we're now in my lab asking whether the age of your cells can impact the age of the organism. So again, in the laboratory itself, in a dish, you can take young cells. We can age them without doing anything other than just dividing them. We can then measure the epigenetic changes associated with cellular aging. And we can then predict whether we're seeing a young cell or an old cell, a cell that's gone through a lot of divisions. And what we found when we look at these types of measures is, again, that they track really well with age in a bunch of different tissues. And to me, one of the really exciting things, at least this was done in mouse, is that we can actually seem to slow the epigenetic age pattern. So in this case, mice who underwent caloric restriction showed slower epigenetic age when looked at in blood. So in talking about the future and how this can actually be used, I think it's important to think about this in terms of we don't each have just one biological age. We have multiple. And in fact, we're actually a mosaic of various biological ages or even epigenetic ages. So essentially every cell in your body probably has its own age. And it's really the pattern of these different ages that's going to determine what your personal disease and health risks are. So are you accelerated on one, but maybe decelerated on another? And what does that mean for your overall health? And the patterns of whether you're accelerated or decelerated are probably due to differences in our genetics, our environments, and our behaviors. So what we found just looking at epidemiological data is that there's actually a number of lifestyle factors that seem to be associated with accelerated versus decelerated epigenetic age when we measure it in blood. So this is everything your mother told you, so no surprises here, but things like physical activity, getting good sleep, eating more fruits and vegetables, having higher education are all associated with decreased or slower epigenetic ages. On the other hand, things like smoking or excessive alcohol use and high stress are associated with accelerated epigenetic age. So the real promise, I think, in the future is, can we actually use these measures to understand personalized lifestyle? So we know that not every lifestyle is going to be as beneficial for every person. We're all different. And so what works for you actually might be based on your epigenetics. So not only are we going to use these to predict perhaps what might be good for an individual, so we can kind of use the algorithms that are used by a lot of these technology companies like Spotify or Waze to actually predict based on your profile compared to other people we've observed what might be best for you. But I also think that people can actually just use these in their own lives, because now we have an outcome that we can actually evaluate whether what we do in our everyday lives is working for us. So with that, I just want to end by kind of reiterating that the technology that we're hoping to develop and currently working on really will give individuals a glimpse into their personalized health and aging. And we really hope that this will enable them to track the impact of their various lifestyles and optimize that to live longer, healthier lives. And with that, I want to thank you and happy to take questions.
And thank you, Morgan. And yes, we have a lot of questions. We're going to start with some of mine, but the audience has also raised a lot of interesting ideas and questions. 2020. So the epigenome, epigenetics are influenced by genetics, as you just said, lifestyle and behavior, which are also affected by our environment. And 2020 has been a year, an absolute standout for its abundance of the negative. COVID, economic turmoil, financial insecurity, political chaos. We all know this list. The election, gut-wrenching exposure of racial and social injustice, the Western states in flames, climate change, writ real, the deaths of Justice Ginsburg. So translating all of that into more sort of behavioral lifestyle changes, we have all through 2020 had extraordinary exposure to stressors, all kinds, large, small, chronic, repetitive, novel, in any case, incessant. Some scientists have actually, behavioral scientists would suggest we're reaching surge capacity for productive coping. Our environment has changed our personal routines and how we are able to practice those things that mom has appropriately told us, how we exercise, how we sleep, how we eat, et cetera, et cetera. There's social isolation, there's loneliness, there's contradictory messaging, politicizing science, leading to fear, uncertainty, confusion. And in these extraordinary times, there's a need for a constant calculation of risk, the smallest decisions, whom I say to hello to, or drop the mask, or if I drop the mask in the park when encountering a friend, can have lethal consequences. So appreciating that these times do not in any way constitute a controlled clinical research experiment. What are your predictions on a population level about how epigenetic profiles may be collectively affected by 2020?
Yeah, so that's a great question. And, you know, as you said, this isn't a controlled experiment, but there is actually so much we can learn from kind of what we might call these natural experiments. So these events that happen that we can then later on trace the impact of. We know that stress has a huge impact on the aging process. I mean, anyone can look at pictures of presidents before they took office and then after 2 terms, and you can visually see the impact that stress might have on the aging process. We know this from animal studies and just from looking at population data. So it's really hard to quantify the degree that 2020 will have in terms of impacting our health going forward, but it'll certainly, especially for some people who are really kind of in the heart of this turmoil, probably have an impact on their aging process. And I think having measures on which we can actually assess that and then see, you know, if we can intervene. I think the other important thing about using measures to track biological aging is you don't have to be perfect, right? You can make one small change. So you can say, I'm stressed out, I have no control over my stress, you know, there's nothing I can do necessarily about this, but what if I tried to, you know, really focus on cooking home cooked meal, you know, I just do one thing for X number of months. And then can you see that small progress one step at a time, rather than feeling completely helpless. So, you know, but I do think, you know, going forward, we will have these large cohort studies that look at kind of whole population data. We're tracking these epigenetic measures in these population studies that are what we call longitudinal. So we're collecting them over time. And we will be able to go back and see if there's a blip for 2020, if all of a sudden people are jumping up in terms of their kind of health risks. And then also if there's biases towards who actually was most impacted. And then hopefully you can go back and actually figure out how do we help certain groups or certain populations to kind of overcome the impact of something like 2020 or what's going on now.
Can one utilize the biologic clock measure? Can one do a test, a retest, another test over the years to calculate the rate of change?
Yes. So we don't actually, you know, the science is kind of new, so we don't know necessarily how often yet people should be doing this. I personally, for kind of an N of 1 experiment, am actually tracking my own epigenetic age every month just to see how stable it is, whether I have, you know, a crazy stressful month, if anything shows up. But yeah, I think, you know, people can track this. And what I would also say is that it's not necessarily important, as important what your epigenetic age is, but it's that rate of change.
Yes.
So can we actually just slow the rate at which we're increasing over time? And that'll have a cumulative effect down the road. So it's kind of this compounding, like deceleration will be much bigger impact later on.
Well, you have a 5-year-old daughter, I understand. So she also confounds the data for you. But while we're curious, we won't ask you how you're doing. So we've agreed that we all know more or less what to do theoretically to, you know, the sort of the common self to live longer, healthier lives. And interestingly, in our expert series, we heard the same advice from many of our scientists, from Dr. Ornish, who literally said, eat well, move more, stress less, and love more. Dr. Tansey, who talks about a shield approach, sleep, handle stress, interact with others, exercise regularly, learn new things, consume a Mediterranean diet. Dr. Barzilai, Dr. Sinclair, of course, concurred, and more. do you think that there might be some advantage to explicitly identifying some of mom's advice as epigenetic interventions that can affect gene expression, and might in fact be information that further assists people in committing to healthier lifestyle?
Yeah, I actually think, one of the most powerful things about these kind of measures is helping people form healthier habits. So it's really hard in terms of aging to say, I'm going to eat really well because in 50 years, it's going to make a difference. I think people have a really hard time having these long-term goals to work towards. So if you say, well, I'm measuring my epigenetic age in a few months, and I really want to make sure I don't mess that up and I want to get a good score, you might actually stick with something. And not only that, is you might actually get more positive reinforcement if you are doing things right. Yeah. As opposed to, even personally, I try to eat a really healthy diet, but sometimes if you're not actually seeing any output from that, it's hard to stick with something and know that it's actually matters what you're doing. I think too, we're innately all optimists. So we always think, oh, well, that won't happen to me. So even if you talk to a smoker, they'll say, well, I know someone who smoked till they're 90 and they're totally healthy. So that's going to be me. I'm not going to be one of the other people. And even thinking about COVID, everyone says, well, it's just those other people who are getting really horribly sick and dying. It's not me. I'm not actually at that big a risk. But I think actually having real, true empirical feedback, I think will help people reanalyze risk. And also, again, work towards, you don't have to be perfect. So, even if you have a diet that might not be perfect, is it better than what it, it's not an all or nothing thing. So I think kind of reframing that and having these endpoints on which we can evaluate our behaviors will be really helpful.
Well, I think what you're describing is assigning a sense or reviving a sense of agency at a time when many of us have felt just a tremendous loss of control. Mm-hmm. And, you have focused primarily on methylation as a measurement of epigenetic change. We know there are other epigenetic chemical changes, hydroxymethylation, histone modifications, influence of microRNAs. Can you say anything about their importance, and will they impact your future research into aging?
Yeah, we study methylation not because we think it's the best epigenetic measure to study, but just because it's the easiest, right? The technology has been developed enough that you're measuring this in thousands of people. You can do it much cheaper. But I think really what we need is to better understand what these epigenetic measures that we're actually capturing are, and how they're regulating gene expression, what they're doing to a cell, and what they're doing biologically. And the only way to actually be able to do that is to layer these other epigenetic information on these other epigenetic marks on top of it, to really, the epigenome is a very complicated thing that even scientists who are at the cutting edge of this don't fully understand what's going on. But the more data we can layer together and understand, both with gene expression data and these other epigenetic data on histone modifications, I think is really where we need to go if we want to figure out what this aging signature is, what we're capturing, and that'll be the only way to figure out, can we actually truly intervene in it? So
I'm going to ask you to bear with me and imagine a scenario. I can't quite myself, but there's a point to the question. So let's imagine a 2-year-old, a 20-year-old, and a 60-year-old who each experience the same traumatic stimulus, would it affect them all to the same degree epigenetically? And I guess what I'm trying to get to is there differential vulnerability to epigenetic influence during different periods of life and development?
Yes, I mean, there's absolutely, our bodies are smart. We've evolved so that we can take in information about our environment around us and somehow modify our biology to compensate for that. And that happens through epigenetics. So that's how we kind of remodel our biology. So, and absolutely, our ability to do that is going to differ depending on probably which life stage we are. But even beyond that, even if you took 5 or 3 6-year-olds, they're not going to all respond the same way. So I think, yes, we have individual differences based on also developmental stage and age, but we also just have innate individual differences based on either genetics or some of the environmental factors that have happened in our past as well.
A similar kind of question, does the accuracy of interpretation of biologic age differ at different chronologic ages? So let me ask that in a different way. I know that you are the bioinformatics advisor to Elysium, a consumer-facing company, and them in their consumer-facing biologic clock called Index. So would utilizing Index be more accurate in a 20-year-old or a 70-year-old?
So I've really worked, both in my research prior to that, but also now that this is going to be a product potentially used by individuals to inform something about their actual life, to make sure that this was accurate for anyone who would take the test. So what we recommend is that this is a test for adults. So we actually find, not to get too mathematical, but the change in this epigenetic pattern is not linear prior to reaching full development, so prior to adulthood. So I would actually say that the epigenetic age measure and how you would interpret it is not accurate for people under age 20, although we can still show that it tracks age, but the number you get is not that meaningful under the age of, for people who are less than 20 years old. However, if we're just talking about adults, we actually find that it's very accurate across the age range. We do find that at the oldest ages, that epigenetic age tends to seem to slow down a bit. So on average, everyone will be quite a bit younger at older ages. And then we also find, so then the other thing to keep in mind is accuracy in terms of predicting future health outcomes. So we're not sure whether, it's better at predicting health outcomes necessarily in people who are 80 or people who are 20. But from what we've seen, we've also, just in my research at Yale, we've tried to split up the data to look at people in different age bins to make sure we can predict things similarly. And so far we haven't found kind of biases towards one age group or another.
What do you see as the next logical evolution of these biologic clocks? What going to look like?
Yeah, so I think, the next key is to get, to actually be able to assess more than one biological age. Because, right now we're using, in general, a clock to represent the biological age of the whole individual. But really understanding these different biological ages and what they mean, and then how together they kind of contribute to the whole profile of an individual, and how that then contributes to, what diseases that person might be more at risk of, or what lifestyle factors might be more beneficial to them based on their kind of this multi-dimensional profile, as opposed to kind of a single dimension biological age profile.
COVID. We have to come back to COVID. We've heard a lot about the genetics of COVID and its invaluable role in helping us increasingly solve many of its mysteries. What role does epigenetics play as we seek to better understand the virus and our vulnerability, potential treatments?
Yeah, so one thing actually my lab is very interested in is whether we can identify differential risk of severe symptoms upon COVID infection. So we know your risk of dying from COVID or even being hospitalized increases significantly the older you are. But it also increases significantly the more diseases someone has. And again, like I was saying earlier in the talk, not everyone who's old is going to have problems if they develop COVID. So can we actually understand whether there's something about the biological aging process itself that makes people more or less vulnerable to COVID? So we're actually doing a lot of work with epigenetics to try and understand if we can actually predict vulnerability. We think that if we can have a measure like this could inform, who in the population needs to, continue social distancing, who maybe can work, who shouldn't, among teachers, who shouldn't be in the classroom, who maybe could, and just a number of other kind of policy planning things involving COVID.
Well, related to COVID, I know that you are well aware, we were talking about stress as a factor in epigenetics, about the concurrent mental health pandemic. Currently, 1/3 of Americans are, according to the national agencies that have this data, 1/3 of Americans are struggling with anxiety relative to same time last year, 1 in 12. 1/4 of Americans are struggling with depression, including former First Lady Michelle Obama, who announced that on her first podcast a few weeks ago. There are reports of increased substance abuse, increased significant suicidal ideation. Many, many years ago, back in the 80s, the brilliant Nobel Prize winner, Dr. Eric Kandel, stated that psychotherapy can be considered a positive epigenetic intervention well before his time. Can you comment on the importance of mental health support as specifically an epigenetic intervention?
Yes, so, just again, stress, especially chronic stress, which a lot of these stressors have become given how prolonged this whole pandemic has basically become, has severe implications on health. And we think also on epigenetics. My lab has looked at a lot of things in terms of kind of pro-inflammatory gene expression associated with either social isolation or social anxiety or depression. And again, we find that this has a dramatic impact on kind of your overall biology. And I think anything, whether cognitive behavioral therapy or whatever it is, that can actually help people handle and, cope with the stress that we really don't have, control over right now, will probably be beneficial. There's such a mind-body connection that, anything you can kind of do to help improve mental health will have an overall beneficial impact on your whole body, so including epigenetics.
Morgan, a slightly different kind of question, ethics. What are some of the potential ethical considerations in epigenetic reporting? Might there not be danger of discrimination from, for instance, life insurance companies and even prospective employers if they knew your epigenetic clock age and rate of aging? Should biologic clock information go into one's medical record I would add voluntarily.
I mean, I would say that, if given how our medical system is set up, if there's any issue that we go back to in terms of pre-existing conditions, then I do not think epigenetic data should ever make it into someone's permanent medical record. I mean, the way I see this is that the epigenetic information should be for the individual. So whether, you want to take a test anonymously. So the nice thing about epigenetics compared to perhaps genetics is we can't actually use your data to identify you. So, someone could hypothetically track their epigenetic aging anonymously, never revealing who they are, and it's for their own benefit and input to assess their own lifestyle things. So it doesn't need to be used by, insurance, whether it's, life insurance, health insurance, or whatever. That being said, I think, in the future, whether, these measures or other aging measures get incorporated into kind of health or promotion and disease prevention in terms of our medical care, I think would actually be beneficial. But again, these are static things. I don't think they should be permanently entered into someone's medical records. And I think too, in terms of something like life insurance, I think it should be optional. If you think you've been doing everything exceptionally well and maybe you should get a, some sort of payback for that in terms of like a life insurance policy where, you think you're a good candidate, then you could voluntarily, I think, use these, but I don't think they should ever be required of people to where they can actually be used against the person to not benefit them.
I hesitate to respond because this is interesting and potentially treacherous territory to navigate, but the point you make about it not being data that can easily identify a subject, unlike many of the directed to consumer genetics testing products. If I don't start asking some of these audience questions, I will never be allowed to handle the audience questions. We have a remarkable number. One of my favorites, imagine. So Morgan, if funding were absolutely no object, what would your dream project be, your most exciting vision to advance the science of epigenetics?
I mean, I think it would have multiple arms. So I think, where we need to go is understanding modifiability. So right now we're kind of just at the forefront of that. So what I would envision would be like a multi-arm intervention where we actually do things like, whether it be a fasting diet or, plant-based diet or different exercise programs, to actually figure out which epigenetic patterns are potentially impacted on those, but also follow people long-term to see if, let's say we change your epigenetic age, does that have implications for your future risk of developing diabetes or heart disease or cancer or whatever it may be. So, I really envision kind of this I'm a data person, so I would just love something on tons of people collecting all the biological data and specimens you possibly can over a very long period of time to really, try and piece all these things together on how they all relate.
So our unnamed philanthropist is not going to ask you what this is going to cost. One of our audience wants to know about the plasticity of the epigenome. Can you reverse changes? That is, I assume what this person means, can one look younger, or is it only possible to slow or alter the rate of decline?
Yeah, so although kind of what I mentioned, these intervention trials are kind of just getting going, there is some very preliminary evidence that you could actually reverse some of this through behavioral interventions. And actually now there's some kind of cutting edge science being developed to actually see if we can use technology or therapeutics to actually do this. But even in terms of looking in animal models, we can see that things like caloric restriction seem to not just slow it, but potentially even reverse it to some extent. So I do think it's possible to do a little bit of reverse thing. That being said, you're never going to have the exact same epigenome you had 5 years ago. You just might have a slightly, something that looks a little bit younger, but it's not the same one you had, in the past.
Morgan, this conversation could continue indefinitely, but again, if I do not send you back to Glenda, mea culpa, it's just been a supreme pleasure, and I know we've just barely touched the surface. This has been fascinating and very valuable, and again, in this time of such chaos and disarray, there is a sense of agency that relates to epigenetics. And I don't want to overstate that or hype that or pseudoscience it, but it's there. And I think of the words, one of the most remarkable things I ever heard George Church say is, don't confuse what's impossible today with what's impossible tomorrow. And you're pushing it, Morgan. You're pushing the edges of it. Thank you. And Glenda, I'll hand it to you for closing.
Thanks both. It was outstanding. Morgan, the first thing I have to say is that we have to have you on again, because you got into such wonderfully complex things, and I think we mostly understand, but we could use a little more help. In any case, I want to ask you some things that may have been covered already, but just a couple of bigger questions. Morgan, can we extend human longevity?
So that's the magic question, right? I believe that we can extend our current median lifespan, so I think most of that's actually going to come from extending the lifespans of people who have more or faster aging, right? So people who probably from lower socioeconomic status, bringing everyone up to a certain level is going to extend the lifespan of the population as a whole. Whether we can extend our maximal lifespan, I'm not taking a position on that yet. I haven't seen any groundbreaking thing that gives me, tons of optimism that we're going to do it in my lifetime. However, kind of like Dr. Stangler alluded to, that doesn't mean we can't, and I'm holding out hope that we can. So I think it's possible, but I'm not sure yet.
Perhaps for your daughter.
Yes, that would be lovely.
And then in that same vein, can our brain span match our lifespan?
Yeah, so that's a really important thing, right? So actually in aging, we talk about not just lifespan, but something called health span. So we don't all want to live to 120 or 130 with failing bodies, and especially not with failing brains. So if we're going to really push the bounds of lifespan so that we're all living, into our mid 100s perhaps, or even into our early hundreds, we need to make sure that our brains are functioning as close to the capacity they do when we're younger as possible. So we don't all want to spend 10 to 20 years in a state of perhaps dementia or cognitive decline. So yeah, I think that's critical.
And is there something about the epigenetics testing or the clock that we can keep looking at? For example, when we talk about brain health, we suggest to people that they start early getting a cognitive assessment baseline, and that's about cognitive performance. But could we, I think you mentioned it earlier, could we get epigenetic testing either annually or every 5 years? And that might be one for example, when you go to the doctor, we're now saying you want to get a checkup from the neck up. But in the terms of epigenetic testing, we're really testing, it's a new kind, could be a new kind of test, and it would be a test for the whole body and brain, I suppose.
Yeah, so I would say that the epigenetic tests that exist today are probably only minimally informative for brain health. But that's where we're hoping to go. We're really trying to develop what are called biomarkers that can assess how your brain is aging without actually having to take a brain sample. So we can do it with a brain sample, but obviously that's not an option for most people. So, you know, we know things like Alzheimer's disease or dementia starts decades earlier. So actually being able to not just monitor that process, but then also use that data to understand how, you know, if you can slow that process or what interventions actually might work. And it also will inform a little bit on the biology of what contributes to brain aging. So I think that's really where we need to go, but we aren't there yet. But I do have some optimism that we're getting a little bit closer, hopefully.
Good. If we, as many aging researchers are thinking, if we make aging a disease and we begin to target it, will many of the chronic diseases that we're concerned about now of aging decrease or disappear?
Yeah, I mean, I think, you know, the whole idea in the field of aging research is that, you know, rather than going after cancer or Alzheimer's disease or heart disease individually, if we just target that biological aging process, so the changes that we know occur as a function of aging that then directly give rise to these diseases. And we don't think that it's, you know, certain changes give rise to cancer and totally different changes give rise to heart disease. And actually, you know, measures like the epigenetic clock actually point to that again, because we know we can use the exact same measure and it's predictive of your risk of all of these diseases. So hypothetically, even though we haven't proven this exactly, we think that if you can reverse or slow those changes, it'll have impacts across the board on all the diseases of aging.
Because aging's the biggest risk.
Yes, exactly.
Just a last small question. I didn't hear you say much about whether there's a pharma intervention on epigenetics or the epigenetic clock or however we say it.
Yeah.
Is there much going on in pharma? Because you're representing a lot of the lifestyle changes, which we're all for. But what about pharma, for example?
Yeah, so I think, you know, I push the lifestyle changes because it's things people can do right now without having to wait for some scientific breakthrough. And there's things you can take control of. I do think there will be, right now, I wouldn't say there is some kind of pharmacological intervention that we know will slow this. There are these large trials that people like Nir Barzilai, and actually I'm involved with his TAME trial, are going to be looking at things like metformin, and we will assess these epigenetic measures and actually be able to say whether they're slowed. And because we're looking at health outcomes, we will actually say maybe we slowed the epigenetic measures, but did that also prevent or slow people's likelihood of developing disease as well? So I think they're on the horizon. We're probably, you know, a few years out from that, but I do think that's what we'll start to be seeing over the next decade.
Fantastic. This was so informative. Thank you so much.
Thank you.
To both of you, to Morgan, you're an outstanding expert for our series. Thank you so much for being here. Ronnie, you did a great job. If we could just understand all the things you're saying. And we thank our audience for the good questions that they've asked. Really, they were terrific questions. And we're glad that our audience is getting updated on the science of brain health. And we thank again to our sponsors, Lugano Diamonds and Alpine Bank, and our friends at Brain Futures and the American Federation for Research on Aging. I want to remind everyone that this session was recorded and will be available on our website, www.aspenbraininstitute.org, in just a few days. Please check our website, sign up for the next episode, and you can as well sign up for the whole series. Thanks, everyone, and ciao. Ciao.
Thank you.
Ciao, Morgan.